Cellular metabolism. Nutrients are any ingested chemical used for growth, repair, or maintenance of the body. There are 6 main classes of nutrients. Water, carbohydrates, lipids, and proteins are considered macronutrients. They must be consumed in large quantities, while vitamins and minerals are micronutrients. They only need to be consumed in small quantities. Carbohydrates are sugars, and sugars are needed for structural components of other molecules, including nucleic acids, glycoproteins, glycolipids, ATP, and related nucleotides. So in A&P classes, and really in all science classes, sometimes when you see larger words, it's beneficial to break it down into its building blocks. So glycoprotein means that it's a molecule that's part sugar and part protein. Glycolipid is a molecule that's part sugar and part lipid. Then you have ATP. ATP is short for adenosine triphosphate, and we'll go through the structure of that and details of that a little bit later in this lecture. And then related nucleotides, GTP, C, AMP, most serve as fuel. They're easily oxidized source of chemical energy. Most cells meet energy needs by a combination of carbohydrates and fats. Neurons and erythrocytes depend solely on carbohydrates. So the base molecule, glucose, serves as the fundamental molecule of energy for the cells, and particularly the brain. So the brain thrives on glucose. Plasma glucose concentration is carefully regulated by insulin and glucagon. Insulin is going to decrease blood glucose. So if you just have a meal, insulin is released, and that's going to take the glucose out of the bloodstream, and it's going to do so in a variety of different ways. It increases the transport of glucose into your cells. It increases the transport of glucose through to the urine in certain cases. It prevents the digestive absorption of glucose. So if you have enough glucose in the body, it's going to help get rid of it. Glucagon is going to do the opposite. So glucagon is going to increase blood glucose during times of need. So if you haven't had a meal for a while, your body and your brain obviously still needs glucose in order to make energy. So glucagon comes in and will get that glycogen that's stored in your muscles and in your liver cells to break down into glucose and mediate that process so that you can increase your blood glucose levels back to a normal range. Hypoglycemia, another one of those big words that you can break apart. Hypo means not enough or too little, and glycemia is talking about the glucose levels of the blood. So hypoglycemia, you don't have enough glucose in the blood. In other words, a deficiency of blood glucose. It causes nervous system disturbances such as weakness and dizziness. So, random side story, my dad got diabetes after COVID. One of those very weird, strange things that happened and came out of COVID. So it's type 2 diabetes. He was on a certain type of medication. They switched him to a different medication, and then he had a knee surgery operation where he didn't really feel like eating after the surgery. He lost a significant amount of weight, and they didn't adjust for that weight loss with his medication. Nonetheless, all of a sudden, he's sitting there one day, and he feels like he's going to pass out. Normal blood glucose levels are in the range of like 80 to 120. His blood glucose levels when he took it at that time was 54. So at 50, you start to have significant neural deficiencies, and you typically pass out at blood glucose of 50. 40 or less, usually you die. So he was at significant risk. So he we called 911 and got him to the hospital, and really all he needed to do was consume sugar. So if my mom would have listened and just gave him a Coke with some sugar in it, or gave him something else with sugar in it, usually like a liquid form of sugar that's easily absorbable, that probably would have increased it, he would have felt just fine. Following that very scary event for my parents, they did talk to the doctor and they switched his medication so it doesn't happen anymore. But that was the first time for them dealing with diabetes for the first time ever in life and having that happen. So lipids, well-nourished adult meets 80 to 90% of resting energy needs from fat, and fat accounts for most of the body's stored energy. Fat is superior to carbohydrates for energy. So fat is hydrophobic and is a more compact energy source substance or storage substance. Fat is less oxidized than carbohydrates, so it contains over twice as much energy. You can get 9 kilocalories per gram for fat versus 4 kilocalories per gram for carbohydrates. So a lot more energy inside of fats. Fat has a glucose-sparing and protein-sparing effect when used for energy needs. Glucose is spared for consumption by cells that cannot use fat, like neurons, and proteins are not catabolized for fuel. Fat-soluble vitamins A, D, E, and K are absorbed with dietary fat. Ingestion of less than 20 grams of fat per day risks a vitamin deficiency. So if you are not consuming fats in the diet, you won't be able to absorb those fat-soluble vitamins. Other functions of lipids, structural, so phospholipids and cholesterol are components of plasma membranes and myelin. Chemical precursors, cholesterol, the precursor of steroids, bile salts, and vitamin D. Thromboplastin, the blood clotting factor, is a lipoprotein. Fatty acids such as arachidonic acid and linoleic acid, precursors of prostaglandins and other iconicides. Important protective and insulating functions. And protein constitutes 12 to 15% of total body mass. 65% of it's in skeletal muscle. Functions would be muscle contraction, motility of cilia and flagella. Structural components of cells, fibrous proteins, globular proteins, and plasma proteins. Buffer pH of body fluids and contribute to resting membrane potentials of all cells. The nitrogen balance. It's the state in which the rate of nitrogen ingestion equals the rate of excretion. Proteins are the chief dietary source of nitrogen. Excretion of nitrogen is primarily through the urine. Positive nitrogen balance means that nitrogen ingestion exceeds its excretion. It occurs in children because they retain protein for tissue growth. Pregnant women and athletes in resistance training show positive nitrogen balance. And growth hormone and sex steroids promote protein synthesis and positive nitrogen balance. Nitrogen does have a bit of deleterious effects on the kidneys. So there's a lot of dietary fads now that say eat only protein and just eat a ton of protein. If you don't follow all of that protein consumption up with a whole lot of water consumption, what can happen is you're going to have too much nitrogen in the blood, which is going to then be converted into urea, and that's going to be filtered through the urinary system at a very fast rate. Now, if you don't have the water to be able to passively get that urea out of the bloodstream, you can have peaks in urea in the blood, but also can end up damaging the kidneys slightly. So it's very important that if you are going to consume an excess amount of protein, that you follow that up with a whole bunch of water. Negative nitrogen balance means that excretion exceeds ingestion. Body proteins being broken down for fuel, muscle atrophy. Muscles and liver proteins are more easily broken down than others. Carbohydrate and fat intake is insufficient to meet the body's energy needs. Glucocorticoids promote protein catabolism in states of stress. So some definitions. Metabolism is the sum of all of the chemical reactions in an organism. Catabolism is when you take larger substances and break them down into smaller substances. So when we talked about the digestive system and how digestive enzymes break substances down into their building blocks, that's catabolism. So when we take something like starch and break it down to glucose, starch is a polysaccharide, glucose is a monosaccharide, that would be catabolism. Similarly, if we start with triglycerides, which are a form of lipids, and break it down into fatty acids and glycerol, that is catabolism. Or proteins broken down into amino acids, again, catabolism. Anabolism is the opposite. So it's taking those building blocks and putting them together to build macromolecules or polymers. So when we consume, let's say we have a salad and we have all different plants that we consume in this salad, and we're going to consume those plants. Well, we don't want to just take the plant material and use it directly to make our own tissues. We don't want to turn into plants. So we would take the plant tissues and we would break it down into the building blocks, because the building blocks are common amongst life. And when we break it down to those building blocks, it's no longer a plant. It's a building block. Then we're going to absorb those building blocks into our bloodstream. The building blocks go to all of the tissues where they can be used, primarily in the liver, and it's going to take those building blocks and build human stuff out of them. So it's going to put them together and make human structural proteins, human structural lipids. It's going to create things that belong in the human body, and then those substances can be transported around the body. So metabolism is a combination of both catabolism, breaking things down, and anabolism, building things. Sources of energy, so from foods, carbohydrates, lipids, and proteins in that order. So our body is first going to use glycogen stores. It's stored in the liver and muscles. Much more glycogen is stored in the muscles than in the liver. Muscles are the primary source of glycogen storage. And it's broken down into glucose. Adipose tissue, fat provides more energy per gram than any other energy source. And muscle mass, protein is used for energy, but only in extreme cases. So if you're under prolonged and extreme stress, or if you have an eating disorder where you're not eating, or you're going through immense periods of starvation, then after the glycogen stores are used and much of the adipose stores are used, you're going to start using muscle mass in order to produce energy. Now, if someone is on the extreme spectrum, and we're talking about an eating disorder such as anorexia, with that, it's not just going to attack the muscle mass of your skeletal muscle. It can also, in very extreme cases, weaken the muscles of the rib cage and slow down respirations. It can weaken muscles, the diaphragm, again, slow down respirations and make things difficult for the body to function, which can eventually lead to death. So definitely want to stay properly nourished and eating and all that kind of stuff. But these are our main sources of energy. So what is energy? Energy is adenosine triphosphate. Adenosine triphosphate looks like this. It's got this adenosine group, and then it says triphosphate. Triphosphate means that there are 3 phosphate groups. Now, where does energy come from here? Energy comes from breaking off this last phosphate. So adenosine triphosphate, is this whole thing, turns into adenosine diphosphate, or ADP. And ADP means that you have the adenosine group and then 2 phosphates. And it constantly goes from ADP to ATP, back to ADP to ATP, and back and forth and back and forth. Now, why does it do this? Because when we break this off, it causes a spark of energy, kind of like throwing gasoline on a piston in an engine. When you put that gasoline in there, it ignites, causing a mini explosion and movement of that piston. And that's what keeps the energy going. And that's how gas kind of works. So if we have this high-charged energy molecule, breaking this off releases energy. And that energy can then be used for some type of movement or for something to happen inside of the cell. When we break this off, it becomes ADP, or adenosine diphosphate, and that molecule is then recharged. A phosphate is added back to it through a number of different processes, and we're going to talk about those processes in this lecture. So the main processes to add a phosphate on to ADP would be the phosphocreatine system, also known as the creatine phosphate system, substrate level phosphorylation, which happens in glycolysis in the Krebs cycle, and oxidative phosphorylation system, which happens in the electron transport chain. So, ATP plus creatine creates creatine phosphate and ADP. Then we have creatine kinase that can take the creatine phosphate, the phosphate from creatine, and add it to ATP. So creatine phosphate is really just storing a phosphate for later use by ADP. When ADP needs to be recharged, creatine phosphate basically donates a phosphate to ADP, and this enzyme creatine kinase assists in that process. That releases creatine and ATP. So because creatine can store phosphates, sometimes bodybuilders will consume excess creatine. The creatine is then stored in muscles, and when they're exercising, this gives them a little bit of extra energy storage so that they can replenish those ADPs a lot faster. The longer you can maintain a muscle contraction, the more the muscle is going to be built. So if you're bodybuilding, you're going to want to have those excess muscle stores so that you can continuously build more muscle. So we will be going through the substrate level phosphorylation and oxidative phosphorylation in just a little bit. So far we've just talked about this creatine phosphate pathway, and basically you've got the creatine with a phosphate. You've got ADP, they bump into each other, mediated by the enzyme creatine kinase, and creatine will lose the phosphate and pass it over to ADP to make ATP. And when it does that, now you've got more energy molecules available for use. So we're going to talk a bit more about enzymes. So most enzymes end in ASE. So as we saw on the previous slide, we have creatine kinase. ACE is an enzyme. All cellular and cellular metabolic processes are regulated by enzymes. So an enzyme is a biological catalyst. What is a catalyst? A catalyst is a substance that reduces the activation energy of a reaction. So that means that naturally this reaction would probably eventually happen. But if we have a catalyst there, it happens faster because the threshold of activation energy, so you have to put energy into the reaction to make it go, that threshold is decreased by the presence of a catalyst. And an enzyme is just a biological catalyst. A substrate would be the substance that is modified by the enzyme. So substrates can be broken down by enzymes, and substrates can be combined by enzymes. It depends on the specific interaction between the enzyme and the substrate. The active site is the place on the enzyme that fits the substrate. Now, we used to call it a lock and key fit, which means that there was an exact shape to the active site on the enzyme, and that exact shape would fit around a substrate exactly. But now we know that it's more of an induced fit model. So what happens is we have the substrate come near the enzyme's active site, and the active site can bend and turn slightly to fit exactly around the substrate. So there's minor adjustments made at the active site to fit the substrate. Now, if the substrate is a completely different structure altogether, it will never be able to fit in that space. So this active site has limited flexibility to be able to fit around substrates because enzymes are very specific to the substrate that they can change. The functional parameters of enzymes, so optimum pH. pH at 7 is neutral. Anything less than 7 is considered an acid. Anything greater than 7 is considered a base or an alkaline environment. So at certain pHs, enzymes are going to function optimally. If the pH for that enzyme is too low or too high, the enzyme can become denatured, or it goes through a process and is denatured. So if it denatures, that means it unfolds and it's no longer functional. So it doesn't completely digest or break down, it just unfolds. And an unfolded enzyme, if you remember learning about primary, secondary, tertiary, and quaternary protein structure in general E&P or in another class, that quaternary structure, that folding of the protein into a functional protein, if it's not folded properly, it's not going to function properly. So a denatured enzyme is a useless enzyme. It's a non-functional enzyme. Now, if it's brought back into the correct pH, it'll fold back up and it'll be functional. But it depends on the pH of the environment of how it's going to fold. And if you think about the biochemistry behind that, it has to do with the side chains of the amino acids that make up the protein. So that's kind of next level biochemistry type detail. Optimum temperature. So there is a particular temperature that enzymes function at. The temperature of the human body, 98.6 degrees. Why is it that temperature, and why do we get sick if it's too high or too low outside of that temperature range? Why do we feel like we don't have a lot of energy when our temperature goes up? Well, too high of a temperature starts to denature proteins. In other words, it's going to unfold those enzymes. And if they're unfolded, they're not functional. So how are we going to produce energy if our body temperature is super high? Not to mention our energy stores are getting depleted by our immune system trying to fight whatever gave us the fever in the first place. But if you think about it, that kind of makes a lot of sense why we're always tired when we're sick, because the optimum temperature for enzymes to function matters. And if the temperature gets too high, those proteins are going to unfold and become not functional anymore. We also have an optimum enzyme and substrate concentration. If you have too much enzyme and not enough substrate, the reaction's not going to continue to go because there's nothing to act on. The enzyme's going to just be there hanging out, waiting for more substrate. If you have too much substrate and not enough enzyme, the enzyme's going to be pretty slow to get through all of that substrate. So there has to be like an optimum concentration or an optimum ratio of enzyme to substrate concentration in order for those reactions to work in the fastest speed. So we're going to look at carbohydrate metabolism, and then we're going to talk about lipid and protein metabolism briefly. Now, with these metabolic pathways, they're much easier to understand in an animation. As I mentioned in class, like we can look at a two-dimensional picture of something and I can talk through a process, but unless you're actually seeing it happen or seeing drawings of the process carried out, it's going to be really difficult for you to understand how the process functions. So in class, I will be drawing this out for you so that you can evaluate all the different pathways and such. Unfortunately, I can't draw in slides that I'm going to be posting due to accessibility laws. So federal mandates and such, I don't have the appropriate technology to be able to alt text every single one of the lines that I draw on a piece of paper. So that's a major limitation of being able to do these drawings and post them online is the accessibility guidelines. But I will be doing this in person in class, which I think will be a lot more beneficial to you because you'll see it happen hands-on. But I'm still going to talk through these slides. Feel free to follow along with this and take some notes. So most dietary carbohydrates, the monosaccharides, glucose, and fructose, are burned as fuel within hours of absorption. So glucose and fructose are our main energy monosaccharides. Those are our main building blocks of energy. Oxidative carbohydrate metabolism is glucose catabolism. So what we do is we take glucose, and this is kind of the very short summary of what happens when we make energy. We take glucose and rip it in half. In that process, we release two energy molecules, and we end up with 2 molecules of pyruvate. Then we take pyruvate and convert it to acetyl-CoA. In that process, we release some carbon dioxide as a waste product. Then we have this acetyl-CoA, and it goes through this cyclical pattern or process called the Krebs cycle. And the Krebs cycle has different names. It's like the TCA cycle, tricarboxylic acid cycle, or citric acid cycle. Whatever you want to call it, the Krebs cycle is kind of my go-to name for it. those acetyl-CoA molecules will go through this process and be modified in a cyclical pattern. This releases 2 more ATP, some carbon dioxide is released, but then energy in the form of electrons and protons are kind of captured from this system. And then we have 2 different carrier molecules that are going to carry these over to the electron transport chain. The electron transport chain, we're going to have some hydrogen ions building up between the membranes of the mitochondrion. That's going to create a proton gradient. We're going to have electrons bounce around in the intermembrane proteins, and eventually those protons are going to flow through this channel-like system called ATPase, and it's going to cause it to spin kind of like a wind turbine. And that's going to cause this gradient to move this pump known as the proton motive force. And as those hydrogens flow through, it provides enough energy to add a phosphate to ADP to create ATP. And that's going to continue to produce about 32 to 36 ATP, and overall the reaction is 36 to 38 ATP per molecule of glucose. So as that happens, a lot of energy is being able to be produced, and we're going to learn or walk through a little bit more of that in detail. So the function of the reaction is to transfer energy from glucose to ATP. So glucose catabolism is accomplished in small steps, each controlled by a separate enzyme, in which energy is released in small, manageable amounts and as much as possible. It's transferred to ATP, and the rest is released as heat. 3 major pathways: glycolysis, anaerobic fermentation, and aerobic respiration. Glycolysis, glucose is split in half into 2 molecules of pyruvate. And then from there, the pyruvate can either go into the anaerobic fermentation or aerobic respiration. If it goes into the anaerobic pathway, it's because we don't have enough oxygen present in the system. When would we not have enough oxygen? Well, if we're doing intense exercise and we're not able to breathe in enough oxygen, if we are under medical distress and we're not able to get oxygen in to the high enough quantities, or if we're doing intense muscle contractions that's constricting blood flow to the area where we're exercising. So that's different reasons where we might not have enough oxygen, but then we'd go through that anaerobic fermentation. But if oxygen is plentiful, we're going to go through aerobic respiration. So the anaerobic pathway, pyruvate would be converted to lactate, also known as lactic acid. This is what makes our muscles burn in an intense workout. That lactic acid doesn't just stay in our muscles. Eventually it diffuses into the bloodstream, gets transported to the liver, where it's converted back to acetyl-CoA and used for energy production later. Aerobic respiration requires oxygen and oxidizes pyruvate to carbon dioxide and water. Coenzymes are enzymes that remove electrons as hydrogen atoms from intermediate compounds of these pathways. Enzymes transfer the hydrogen atoms to coenzymes. Coenzymes donate them to other components later in the reaction pathways. 2 coenzymes that are really important are NAD and FAD. NAD can be converted to NADH, and FAD can be converted to FADH2. Hydrogen atoms removed from metabolic intermediates in pairs, so 2 protons and 2 electrons. They're transferred to a coenzyme. This produces a reduced coenzyme with a higher free energy content than before the reaction. The coenzyme becomes temporary carriers of the energy extracted from glucose metabolites. FAD binds 2 protons and 2 electrons to become FADH2. NAD binds 2 electrons, but only one of the protons to become NADH. The other proton remains free in hydrogen, or remains a free hydrogen ion. Glycolysis is a metabolic pathway that splits a glucose molecule into two molecules of pyruvate. So they break it down into major steps here. The textbook lists out all of the independent steps. So hexokinase is an enzyme that transfers an inorganic phosphate group from ATP to glucose. It produces glucose 6-phosphate. This is an extremely important step because glucose can diffuse down a concentration gradient. So let's say we have a lot of glucose out of the cell. Well, glucose is going to diffuse down its concentration gradient. It's usually brought into the cells through some type of facilitated transport process or through an ATP sodium pump, some type of facilitated process through a protein. And once it gets into the cell, it's going to float down its concentration gradient until there's an equal concentration inside and out of the cell. Well, what we don't want to happen is we don't want the glucose to stay outside of the cell once it reaches that equilibrium. We want to keep bringing more and more glucose into the cell. So how do we do that? Well, once glucose is in the cell, we turn it into something that's not glucose. So then more glucose will come into the cell. So this first process really traps glucose in the cell, and it traps it as glucose 6-phosphate. All it really does is it adds a phosphate group to glucose. It keeps intracellular concentration of glucose low, favoring continued diffusion of glucose into the cell. It prevents sugar from leaving the cell since phosphorylated compounds cannot pass through the membrane. G6P can then be converted to fat or amino acids, polymerized to form glycogen, or oxidized to extract its energy. Oxidized means that it would be going through the next step of cellular respiration. So here's all of those steps kind of laid out in order. You can take a moment to pause and review the diagram, and you can also look up the animations that I've posted to watch videos of the process. Then we have priming, so G6P is rearranged to form fructose 6-phosphate, phosphorylized to form fructose 1,6-diphosphate, primes the process by providing activation energy, and 2 ATPs have been consumed. Cleavage. Fructose 1,6-diphosphate lyses or splits into 3 carbon molecules. Small changes result in 2 molecules of phosphoglyceraldehyde, or PGAL, also called glyceraldehyde 3-phosphate. Then we have oxidation. Each PGAL molecule is oxidized by removing a pair of hydrogen atoms. The electrons and 1 proton are picked up by NAD+, yielding H+, and, this is all kind of an NADH. Phosphate group is added to each C3 fragment from the cell's pool of free phosphate ions. Dephosphorylation happens. The phosphate groups are taken from glycolysis intermediates and added to ADP, making ATP. And then the C3 compound becomes pyruvate. The end result of glycolysis. 4 ATP are produced, but 2 are consumed. So that's a net gain of 2 ATP per glucose. Some of the glucose's original energy is in the ATP, some is in NADH. Some is lost as heat, but most of the energy remains in pyruvate, and the end products are listed here. So if there's not enough oxygen, that pyruvate is then going to be going down the fermentation pathway, or the anaerobic respiration pathway. In the absence of oxygen, or mitochondrion, cells can only generate ATP through glycolysis. This is not enough to sustain a human life, but it can get us by for a few minutes. Glycolysis cannot continue without a supply of NAD. Anaerobic fermentation, NADH donates electrons to lactate and regenerating NAD+. Lactate leaves the cells and goes to the liver. When oxygen becomes available, the liver oxidizes it back to pyruvate. Oxygen is required for this part of the reason, this is part of the reason why we breathe more vigorously after exercising. Liver can also convert lactate back to G6P and polymerize it to form glycogen for storage, or remove the phosphate group and release free glucose into the blood. Limitations: it's wasteful. Most of the energy of glucose is still in the lactate and has contributed no useful work. Lactate is toxic. It has to be metabolized in the liver. Skeletal muscle is relatively tolerant of anaerobic fermentation, cardiac muscle less so. The brain empties, no anaerobic fermentation. The brain employs no anaerobic fermentation. In the presence of oxygen, pyruvate enters the mitochondria and is oxidized by aerobic respiration. Most ATP generated in this way the most ATP is generated in this way. It requires oxygen as a final electron acceptor. Sometimes you're going to see test questions that say, what's the terminal electron acceptor for aerobic respiration? It's oxygen. It occurs in two practical steps. The matrix reactions, their controlling enzymes are in the fluid of the mitochondrial matrix, and membrane reactions, their controlling enzymes are bound to the membranes of the mitochondrial cristae. The matrix reactions. So 3 steps prepare pyruvate to enter the citric acid or Krebs cycle. Decarboxylation converts C2 compound to acetyl group or acetic acid, and acetyl group binds coenzyme A. It results in acetyl-CoA. Acetyl-CoA is formed after that process of glycolysis. Then acetyl-CoA can enter into the Krebs cycle, also known as citric acid cycle or TCA cycle. The matrix reaction, so acetyl-CoA combines with C4 oxaloacetic acid to form citric acid, and that's the start of the citric acid cycle. Water is removed and citric acid molecules are rearranged. Hydrogen atoms are removed and accepted by NAD. Another carbon dioxide is removed and the substrate becomes a 5-carbon chain. Previous step repeated, removing another free carbon dioxide, leaving a 4-carbon chain. Some of the energy in the C4 substrate goes to phosphorylate guanosine diphosphate, or GDP, and converts it to guanosine triphosphate. 2 hydrogen atoms are removed and accepted by the coenzyme FAD. Water is added, 2 final hydrogen atoms are removed and transferred to NAD. The reaction generates oxaloacetic acid, which is available to start the cycle again. This is why it's considered cyclical. So this is a summary of the matrix reactions. We end up with 2 ATP in the end of this. So we made 2 in glycolysis, now we made 2 in the Krebs cycle. Again, not enough to sustain life. We need 1 more additional step. The carbon atoms of glucose are carried away as carbon dioxide and exhaled. So when you're asked questions like, why do we breathe? Well, we have to bring oxygen in to serve as a terminal electron acceptor for the electron transport chain. Why do we breathe out? To eliminate carbon dioxide. Carbon dioxide accumulates in the tissues, and then it goes through, we'll learn about the acid-base balance of the body, but it gets converted to bicarbonate, transferred through the bloodstream to the lungs, where it's exhaled from the lungs as carbon dioxide. So the membrane reactions have 2 purposes. They want to further oxidize NADH and FADH2, and then regenerate those carriers for later on in the process. The electron transport chain is a series of compounds that carry out the series of membrane reactions. Most of them are bound to the inner mitochondrial membrane, arranged in a precise order that enables one to receive a pair from the electrons from the member on one side of it, pass electrons to member on the other side. These are the molecules that are embedded in the membrane. It's sufficient to kind of review the names and be familiar with the names. You don't need to know all of the details of what they do. And we continue. So hydrogen atoms are split apart as they transfer from coenzymes to the chain. Protons are pumped into the intermembrane space. The electrons travel in pairs along the transport chain. Each electron carrier becomes reduced when it receives an electron pair and oxidized again when it passes the electrons along to the next carrier. Oxygen is the final electron acceptor. So each step along the way, electrons are transported, and they build up that proton gradient in the intermembrane space. The chemiosmotic mechanism. So the electron transport chain energies fuels respiratory enzyme complexes. Inner membrane is permeable only to hydrogen ions, only at channel proteins called ATP synthase. Chemiosmotic mechanism, current rushing back through the ATP synthase channels drives ATP synthesis. So here you can see the electrons are going to bounce around in these different complexes. Protons or hydrogen atoms are going to be pumped into this intermembrane space. So you can see the hydrogen ions here. And they try to get back through down their concentration gradient, but the only place they can get back through is through this special rotating pump called ATP synthase. Now, when they go through, the force of them traveling through is going to cause this to rotate, and that gives enough energy to take ADP, add a phosphate to make ATP. This is an overview of ATP production. So overall, it produces ATP. So the complete anaerobic oxidation of glucose to CO2 and H2O produces 32 ATP. And here you can see where all of that occurs. This is a really useful diagram. I know there's some summaries provided in the assignment stuff, but definitely pause the video, take a look at everything that's happening. So glycogen metabolism. Glycogen is the energy storage molecule. It's stored in your muscles and liver. ATP is quickly used after it's formed. The body converts extra glucose to other compounds better suited for energy storage. Glycogenesis. Glyco is talking about glycogen. Genesis is talking about the creation of something. So glycogenesis is the creation of glycogen, or the synthesis of glycogen. It's stimulated by insulin. So if there's too much blood sugar, or too much sugar in the blood, we want to reduce the amount of sugar that's in the blood. And how do we reduce it? Well, combine it together into the storage form of glycogen and store it in your muscles and liver for later use. Glycogenolysis. Glycogen is written out in there. That's referring to glycogen. Lysis means to cut up or break apart. So glycogenolysis is the hydrolysis of glycogen, or the breakdown of glycogen. Hydrolysis is talking about hydrolysis. Hydro is referring to water. Lysis is to break apart. So this is breaking down glycogen into its building blocks using water. Stimulated by glucagon and epinephrine, liver cells can release glucose back into the blood and provides glucose between meals. Gluconeogenesis. Gluconeogenesis. So talking about glucose, neo is new, and genesis is the creation of. So it's the synthesis of glucose from non-carbohydrates, or creating new glucose from substances that are not carbohydrates, such as glycerol and amino acids. It occurs chiefly in the liver and can also occur in the kidneys if necessary. So take a minute to pause the video and review this diagram. Okay, so I already kind of talked about this a little bit, but glucose metabolism is regularly controlled, or relatively mostly controlled, by insulin and glucagon. Insulin, again, decreases blood glucose levels through a variety of different processes, while glucagon increases blood glucose levels primarily by increasing the amount of absorption of glucose from the diet, decreasing the amount of glucose that is shed in the feces. We would then increase glycogenolysis, so breaking down glucose to release that into the bloodstream. This is a really useful diagram that shows you the relative levels of glucose. So normal blood glucose levels, as I mentioned earlier, would be between like 80 and 120, and anything below 40 is indicative of, someone could die if it drops below 40. 50 is usually where consciousness is lost and you have the neurological symptoms associated with hypoglycemia, and then like we always want to try to see it between that 80 to 120 range. You can see how glycogenolysis would break down glycogen to release glucose into the bloodstream to try to increase blood glucose. You can also see gluconeogenesis, where the liver would mediate the conversion of lipid molecules or amino acids to glucose. So non-carbohydrate sources converted to glucose to increase those blood glucose levels. With hyperglycemia, the opposite end of the range is when it's too high of blood glucose. So that's if it gets really high, glucose can spill over and get excreted into the urine. That would likely indicate more of a diabetic state. If it's at about like 140, that's when you have fat formation. So you have so much glucose in the body, it's actually not only going to store it as glycogen, but also as fat. And then if you have the normal regulatory amount of glucose, digestive system, you would break down the carbohydrates to glucose, it would get absorbed, and you'd have about 100 mg per deciliter of glucose. And then you have glycogenesis, so you would create glycogen in the liver, and that's how you would store it in the liver. Or you could break it down and use it for energy through those catabolic processes that we've been talking about this lecture. This is a master diagram that kind of shows how all of the pathways are related. So the carbohydrate pathway is here in the middle, but the carbohydrate pathway is linked to the lipid pathway and protein pathway. So take a moment to pause the video and look through this. The homework assignment has a lot of these pathways put together, but you can kind of take some time to just internalize this and see how substances would move through here, where proteins could enter into the citric acid cycle, and where lipids could enter into the carbohydrate pathway as acetyl-CoA, or it could leave the carbohydrate pathway as ketone bodies. And we'll talk more about ketone bodies here in just a little bit. But in general, if you're going to be using lipids for energy production, so let's say you have a carb-free diet and you cut out all carbs from your diet, well, the next most energy-rich source from the diet would be lipids. So if you're using lipids for energy production, lipids are going to eventually cause the formation of these ketone bodies, and it could lead to ketoacidosis. Now, if you're diabetic, type 1 diabetics typically can't utilize glucose because there's no insulin being produced, so you have to have insulin injections. And with that, either the insulin is produced as defective or not at all. And or there's like an autoimmune attack on the insulin producing cells of the pancreas, so you're not producing enough for the right type of insulin. And when that happens, diabetics are kind of forced to use lipids for energy production, and that's where you can have those ketone bodies formed, which can cause ketoacidosis. So lipids, there's continual turnover of lipids in the body as they are synthesized for energy storage, lipogenesis. Again, if you break down these parts, you can deduce the definition from them. So if you're asked a question on an exam and it says, what is the creation of lipids known as? Well, that's lipogenesis. Genesis is the creation of or synthesizing, and lipo is talking about fats. This is different from glycogenesis, which is talking about the creation of glycogen. Lipolysis, or lipolysis, is breaking down fat. Lysis, again, is breaking things down. So lipo is referring to fat, so that would be the breakdown of fat. And here's the pathways of lipolysis and lipogenesis. And you can take a moment to kind of look through this diagram here. So lipolysis forms glycerol and fatty acids, and it's stimulated by many hormones: epinephrine, norepinephrine, glucocorticoids, thyroid hormone, growth hormone. Beta oxidation in the mitochondrial matrix catabolizes the fatty acid components. A fatty acid with 16 carbons yields 129 molecules of ATP, definitely a richer source of energy than the glucose molecule. Fatty acids catabolized into acetyl groups by beta oxidation in the mitochondrial matrix may enter into the citric acid cycle as acetyl-CoA, or under ketogenesis, it's metabolized by the liver to produce ketone bodies. The ketone bodies are called acetoacetic acid, beta-hydroxybutyric acid, and acetone. The rapid or incomplete oxidation of fats raises blood ketone levels, or ketosis, and may lead to a pH imbalance, ketoacidosis. Diabetics then that experience this, and even people who are just doing a carb-free diet and relying excessively on fats for energy production, if they go through ketoacidosis, they would have like an alcohol smell on their breath possibly, and their pH would be super low in the blood, denaturing a lot of their enzymes and causing things to not function appropriately. People can get pretty sick and die of ketoacidosis. So proteins. Cell draw upon the amino acid pool to make new proteins. Dietary amino acids plus 100 grams of tissue protein broken down into each day into free amino acids. Some amino acids in the pool can be converged to glucose or fat directly used as fuel. Conversions involve three processes: deamination, amination, and transamination. If you remember the typical structure of an amino acid has an R group, so it's a group that's very specific to making it the type of amino acid that it is. And then it has a COOH carboxyl group, and then it has an NH2 amine group. So deamination is just ripping off that NH2 amine group. Amination would be adding an NH2 group. And transamination would be transferring an NH2 from one molecule to another. Proteins used as fuel. To be used as fuel, amino acids must first be deaminated. So you have to take off the NH2. What remains is a keto acid. It may be converted to pyruvate, acetyl-CoA, or one of the acids of the citric acid cycle. All of these reactions are reversible in case there's a deficiency of amino acids. In gluconeogenesis, keto acids are used to synthesize glucose. Transamination, ammonia, and urea. So when an amino acid is deaminated, glutamic acid is formed. Amino group of amino acid is transferred to the citric acid cycle intermediate. Glutamic acid can transfer to the liver. NH2 is removed, converting it back to alpha-ketogluteric acid. NH2 then becomes NH3, which is toxic and can't be allowed to accumulate. So it enters into the urea cycle, and that's the pathway by which the liver combines ammonia with carbon dioxide to produce the less toxic waste molecule urea, and urea is then excreted in the urine. Protein synthesis we're going to go through in detail in a future lecture, so I won't say much about this, but it's a process involving DNA, mRNA, and tRNA and ribosomes, stimulated by growth hormone, thyroid hormone, and insulin. It's known as transcription, and then followed by translation. So proteins, this is just another diagram of the deamination. So proteins undergo deamination, producing ammonia, which is converted to urea, and keto acids, which are converted to glucose through gluconeogenesis. Keto acids may also be converted into pyruvic acid or acetyl-CoA, which enter the citric acid cycle. Glucose enters glycolysis, and urea is excreted as waste. So you can take a minute to pause the video and review this diagram. So we talked a little bit about the liver before, but the liver plays a wide variety of roles in carbohydrate, lipid, and protein metabolism. Most of its functions are non-digestive. Hepatocytes perform all functions except phagocytosis. They're not able to eat things. Degenerative liver diseases such as hepatitis, cirrhosis, and liver cancer are especially life-threatening. Metabolic states. So body metabolism changes depending on time since last meal. There's the absorptive or fed state, which happens about 4 hours during and after a meal. Nutrients are being absorbed. Nutrients may be used immediately to meet the energy and other needs. Post-absorptive or fasting state prevails in the late morning, late afternoon, and overnight. Stomach and intestines are empty, and the body's energy needs are met from stored fuels. This is where the fasting diet, and they've been talking about intermittent fasting a lot lately, but that's where this comes into play. So I'm not going to get into too much of intermittent fasting, but it is a dietary strategy where you would basically keep the body in a fasting state for a longer period of time, which forces it to break down stored nutrients in the body to use that for energy production. And the longer you can hold out without having those stores, then the more you're going to be utilizing the stuff that's already in the body. Now, there are some very big risks with this. Nutrient deficiencies are a huge risk. We never want to starve ourselves. Then definitely if you are already kind of within the normal weight range, losing an excessive amount of weight would not be a good thing. But that's just one dietary strategy and kind of how it works. So during the fed state, glucose is readily available for ATP synthase. It serves as a primary fuel and spares use of stored fuels. Carbohydrates during the fed state, absorbed sugars are transported to the liver by the hepatic portal system. Most glucose passes through the liver and becomes available to cells everywhere. Glucose excess is absorbed by the liver, which forms glycogen or fat, and most fat is synthesized by the liver, is released into the circulation. Fats enter the lymph as chylomicrons. Lipoprotein lipase removes fats from chylomicrons for uptake by tissues, and fats are primary energy substrates for hepatocytes, adipocytes, and muscle cells. Amino acids during the fed state most pass through the liver and go on to other cells for protein synthesis. In the liver, they may be used for protein synthesis, fuel, or for ATP synthesis or fatty acid synthesis. The absorptive state. So regulated by insulin and several intestinal hormones, insulin regulates glucose uptake by nearly all cells except neurons, kidney cells, and erythrocytes. It increases the cellular uptake of glucose, causing plasma glucose concentration to fall. It stimulates glucose oxidation, glycogenesis, and lipogenesis. It inhibits gluconeogenesis. It stimulates active transport of amino acids into cells and promotes protein synthesis. And it acts on the brain as an adiposity signal, or an index of fat stores. The post-absorptive state regulates plasma glucose concentration to between about 90 to 100 mg per deciliter. Especially critical to the brain, uses stored fuels as needed. Carbohydrates during the fasting state, glucose is drawn from glycogen reserves or synthesized from other compounds. Liver usually stores enough glycogen to support 4 hours of post-absorptive metabolism before gluconeogenesis occurs. Fats during the fasting state, adipocytes and hepatocytes hydrolyze fat and convert to glycerol to glucose. Free fatty acids cannot be converted to glucose, but they can favorably affect plasma glucose concentration by having a glucose-sparing effect. Free fatty acids are oxidized by the liver to ketone bodies, which other cells absorb and use as a source of energy. Proteins during the fasting state are used as fuel when glycogen and fat reserves are depleted. Cachexia is extreme wasting away seen in some chronic diseases resulting from a loss of appetite, anorexia, as well as altered metabolism. So there's a lot of text and a lot of information here, but just try to, in your mind and in your notes, differentiate the absorptive state and the fasting state. In the fasting state, you need to somehow find a way to get glucose back into the blood. So what are the processes that can happen that will increase glucose in the blood? During the absorptive state, you're bringing glucose into the blood, so you have to find a way to decrease the blood glucose levels. And you're going to do that by shuttling the glucose into the cells, by using the cells to convert glucose to fats, or breaking it down to make energy. So if you follow like a logical thought process to try to diagram and compare the post-absorptive fasting state to the absorptive state, it'll make a lot more sense for you. So definitely recommend like diagramming this out. Regulation of the post-absorptive state, regulated mainly by sympathetic nervous system and glucagon. As the blood glucose levels drop, insulin secretion declines. The pancreatic alpha cells secrete glucagon. Regulation continued. Sympathoadrenal system also promotes glycogenesis and lipolysis. Cortisol released in response to stress, which promotes fat and protein catabolism and gluconeogenesis. The metabolic rate, the amount of energy liberated in the body in a given period of time. It can be measured directly with a calorimeter, closed chamber with water-filled walls that absorb heat given off. It's measured indirectly with a spirometer by measuring the amount of oxygen a person consumes. It depends on physical activity, mental state, absorptive or post-absorptive status, thyroid hormone, and other hormones. The basal metabolic rate, or BMR, is the baseline that minimizes the effects of activity, feeding, and hormone levels. Relaxed, awake, fasting, comfortable at room temperature, adult male BMR is 2,000 kilocalories per day and slightly less for a female. The total metabolic rate is the sum of the BMR, basal metabolic rate, and energy required for voluntary activities. Factors that raise the TMR would be physical activity, pregnancy, anxiety, fever, eating, catecholamines, and thyroid hormones. Factors that lower it would be apathy, depression, or prolonged starvation. As one reduces food intake, the body reduces its metabolic rate to conserve body mass, making weight loss more difficult.